Okay, Assalamualaikum and good morning. Okay, I would like to welcome all of you to the 8th session of Breakfast at FOM for this year. I'm Associate Prof. Dr. Zamri, Chik, Head of Pharmacology Department. So will be a moderator for this session. So of course, first I would like to thank the organizer for giving this opportunity to Pharmacology Department. And we are so delighted today that we have a young lady, very beautiful young lady this morning, a senior lecturer from Pharmacology Department, Dr. Sharifah Zamiah, Syed Abdul Kadir. Okay, she is actually just some introduction about her. She is a UK trained scientist, actually recently completed her PhD from the University of Manchester with an interest in cardiovascular research. So her expertise is actually in microvasculature physiology investigation techniques. So it's very keen to share her research with all of us today. And she is going to talk about oxidant, either it is good or bad to our vascular system. Okay, this is very interesting topic. So without further ado, I just would like to invite Dr. Sharifah to give a talk. But before that, if you have any questions, okay, for her, just actually use the Q&A box and we will read at the end of the session. Okay, Dr. Sharifah, the floor is yours to start.
Thank you so much, Dr. Zamri, for the introduction, and good morning, everyone. And thank you for being patient as well for waiting after we start our all-day technical issue. So today I'm going to talk to you about oxidant and the microvascular system. So is it a friend or a foe? I'm just going to continue where I left off about the key 5 key things that I'm going to deliver today. First, I'm going to emphasize on the importance of microvasculature. Second, I'm going to share with you a novel mechanism pertaining small artery dilation. Third, it's the potential of, it's just a quick sneak peek of the potential antihypertensive agent that we recently discovered. And also, this is a bit of a personal vendetta, not vendetta, but a personal goal for me, is that I would like to show the new technology that we have here in the Department of Pharmacology for everyone to take advantage of. And 5 is just a little sneak peek of the future adventure of the vascular research in UM and how I would, would like to direct that. So we're gonna, as the introduction, we're gonna talk about resistance artery. When you first hear about the vasculature system, when you first hear about the vasculature system, this image would pop in mind. You know, it's highly vascular, vasculated. But let's take a look at the, I don't know why it's moving on its own. So let's take a look at the small resistance artery here. It plays such an important function compared to the rest of the organs because, first of all, it provides a resistance against the systemic blood pressure. To protect constriction of small resistant blood vessels actually determine your blood pressure. And second of all, these artery right here also have such a big burden whereby it has to regulate flow, the pressure, the volume, and to ensure that the optimum amount of oxygen and also nutrient is being delivered to the downstream capillary bed. So, and I would like to emphasize on the key features that are only present in the small resistance artery and have not been found in other big conduit arteries. So first of all is that small resistance artery will constrict in response to pressure. So this is actually a protective mechanism to protect the capillary beds. And second of all, there is a signaling hub, which is called microendothelial projection. So we're going to talk about this furthermore. And third of all, it is highly innervated. So this is why small resistance artery are able to respond very quickly within milliseconds to seconds to meet the demand of the organs. Okay, and so we're going to talk about, so in small resistance artery, endothelium is actually the key regulator in vascular function. So it is well known that nitric oxide dilates artery, but that is a dominant mechanism within big blood vessels. So as for small resistant arteries, small blood vessels, it is always driven by EDHF, endothelial dependent hyperpolarization factor. So a little bit more on the endothelium is that it is a monolayer cell and it covers the whole cardiovascular system, and its function as a barrier and also mainly vasodilation. And endothelium of small heart artery modulates vascular cell calcium, and it is such a critical, it's critical for the control of the blood tone, pressure, and volume like we discussed earlier on. So if you take a look at this diagram here, this is what I meant by the distinct feature of small resistant artery. You can see that there's a invagination of the endothelium through the internal elastic lamina, and it is anchored. It's anchored directly onto the vascular smooth muscle cells. And if you take a look at this diagram, it does not just anchor, but it is also connected via gap junction. So these features in small resistance artery, or solely to small resistance artery, is the one that allows this transmission of hyperpolarization through the vascular smooth muscle, which allows it to dilate. Okay, so now we're going to go through a little bit of mechanistic in order for you to understand the findings. So I'm going to take you through a key regulatory pathway. So this is a well-conserved pathway. It's well established in small resistance artery. So we have here the activation of muscarinic receptor, which activates the GQ-PCR protein and activates a membrane-bound protein called protein lipase C. And protein lipase C would cleave phosphatidyl inositol 4,5 biphosphate on, and the glamorous name is PIP2. So PIP2 will give you two, and it will further hydrolyze and cleave and give you two very important second messenger. The first messenger is IP3, referring here. So this one it plays an important role in regulating intercellular calcium storage, the endoplasmic reticulum, whereby DAG activates other. So in this case, it activates an anchoring protein. So this anchoring protein is a constitution of an A cap and protein kinase C. So when it is anchored together, it activates a very powerful ion channels, which is called TRPV4 ion channels. So TRPV4 ion channels, it stands for transient receptor potential valenoid 4. So it is actually a calcium permeable ion channel, selective to cation channels. Okay, so it is due to the electric coupling here. So when there is an increase in a localized calcium, so this is not a global increase, yeah? This is a localized just in the MEP, it will activate all these other channels that would lead to hyperpolarization. Hyperpolarization of the endothelial transmitted to the vascular smooth muscle will lead into dilation. So you will see this diagram along the slide. So it's ACH, activates PLC, activates PIP2, and two important messenger, IP3 calcium release, and also TRPV4 calcium influx. This would increase the calcium concentration within the myoendothelial projection, and that causes activation or IKSK channel, aka that is also referred to EDHF, the endothelial hyperpolarization factor. Okay, so where does oxidant fit into the picture? So now that we know the physiology of small resistance artery, I'm going to introduce you to a new activation pathway of protein kinase G. Okay. So sorry. So protein kinase G has been around for like 60 years since it's been discovered. And it is known as the abundant protein in the vasculature. Its vital role is for the relaxation of vascular smooth muscle cell that is well defined in the literature. It is found in both endothelial and smooth muscle cells. However, within the endothelial, the function of protein kinase G, as we know it, the traditional function activated by CGMP is so ambiguous. There are some evidence showing that it constricts the vessels, and there are some evidence that dilate the vessel, but it always go back to which vessel that you're looking for, that you're looking at. So, however, recently, less than a decade ago, our collaborator in King's College London found out that there's a new mode of activation of protein kinase G. So if you take a look at the diagram here, where am I? Sorry. Oops. All right, there. So the diagram here, you can see that this is the classical activation. So it is phosphorylated upon activation by CGMP. And this is the new discovered pathway whereby, when there's an oxidant, there'll be a formation of a disulfide bond at cysteine 42 location of the protein. So why am I telling you all this very in depth? It's because we have generated a genetically modified mice whereby all PKG in the system cannot be activated by oxidant. So it is silent. So this pathway right here is silent. So one of the things that during the collaboration that we noticed, and upon adolescence, these animals actually develop hypertension automatically, and it gets worse within age. And does that sound familiar to you? It's just like a human being, isn't it? Like it gets worse with age. So we developed this genetically modified animal whereby we change, instead of cysteine, whereby oxidant binds there and form a disulfide bond, we knock it out and have serine. So there's no activation by oxidant, and hence no disulfide bond, so it cannot phosphorylate the target. Okay, so so you must be asking like what oxidant are you referring to then? So the oxidant, so this is a research by our team whereby we have managed to identify the oxidant is actually coming from the interluminal pressure. So just a quick recap that with interluminal pressure sensed by the mechanistic sensor, it produces ROS. So this ROS actually activates the PKG dimer. So and we did the experiment detecting this in vascular smooth muscle. So how about endothelial? Because endothelial plays such an important, such a vital role in dilating small arteries. So today I'm going to share to you, is it a friend or a foe? What does it do then to the endothelial function? And how do we, before we get there, I just want to show you one of the key things that I want to share with you today is the new technology. So what are you looking at the moment right now is pressure myography technique. And this is recently being it's been gifted by University of Manchester to us to run the research here. So just briefly, we take a mesenteric artery. You can see here, we take a third order, which is close to the ileum, mounted with a cannula. So once it's mounted, we pressurize it and it will come out with the graph. So for the graph, we have the time of the experiment and also the diameter, okay? And I'm gonna take you into the lab with me right now, and by watching a video from our team member that's recording this. So in this video, I want you to pay attention on the artery. Take a look at the size of the artery. So just a quick, the parameter here that's been recorded is the diameter. This is the temperature. We always make sure the temperature is maintained according to the physiological temperature. And here is pressure. So when we introduce pressure, you can see it goes down. So do you remember that I say about one of the key features of small resistance artery is it constricts in response to pressure. So this is the constriction that I meant when it responds to pressure. Imagine there's no pharmacological agent in there, just pressure, and it constricts. So this is the protective mechanism to protect the delicate capillary beds. So I'm going to go through the experiment here. So do you remember this mechanism that I mentioned earlier? We're going to start off with activating ACH. So wild type is the healthy animal, whereby PKG is the animal that cannot, that PKG cannot be activated by oxidant. So you can see when we activate, when we introduce acetylcholine at 10 micrometer, at 10 micron, you can see macromolar, sorry, at 10 macromolar, you can see approximately 90% of dilation, compared to calcium-free. So calcium-free will give us a passive diameter of the artery, like the whole maximal dilated diameter. But if you take a look at here, when PKG cannot be activated in this animal, even when you introduce a maximum dose of 10 micromolar of acetylcholine, it only dilates up to like less than 20%. So there's two important key things that I want you to take from the slide. First of all, do you remember when I told you that ACH dilation in small artery is a highly conserved regulatory pathway? So this conserved and vital pathway is silent when PKG cannot be activated by oxidant. So it says that oxidizable PKG, it never thought to have such critical and such important role in the endothelial function, but now we know. But the next is to verify that what we see by bypassing all this mechanism and activate the IKSK channel. So when we see the dilation here, both of the animals, it dilates to NS301, NS309. So this is actually a compound that targets IKSK channel. Which means that, you know what, they are still capable of ADHF respond. However, there are something wrong, upstream of the pathway. So there's something wrong here in the pathway. So now within the pathway, we're going to look into the TRPV4 calcium influx. Okay, in order to do this, I activate the TRPV4 calcium channel directly with a compound called GSK101. So if you take a look at here, look at the diameter, whereby we introduced 10 nanomolar of GSK101, a direct activator of TRPV4. We can see, up to 90% of dilation, which means that the channel is function, correctly. But here at 10 nanomolar, they're, only around, less than 20% dilation as well at 10. However, when we challenge the artery by giving 10 times more, I know it's not physiological, it's just that we want to see, if the channel is functioning. And to our surprise, there is a dilation at 10 times those more. So what does this tell us is that there is an antagonistic effect of PIP2. And also, if you remember that I talked to you about, in order to activate TRPV4 channel, you have to have the connection, the anchoring protein, the DAG, PKC, and ACAP protein in order to activate. So we think that it is, upstream. Oops, here. However, before we just jump into the conclusion that it got to do with PIP, we have to verify it. So when it comes to novel mechanism, it's always verification of the verification to sell the idea. So this is actually a cutting edge technology where we're using a spinning disc confocal microscopy, whereby this microscope, it will take up to 60 images per second. So every millisecond is one image. So that allows us to see, fast transient response like calcium movements. So we're using this technology, and my professor in Manchester always call it the Lamborghini of the team, because it does, it's the same price as a Lamborghini. So taking you through the procedure, we still take the third order mesenteric artery. So we cut it, we slide it through, and have it at an on fast preparation. On fast meaning that the endothelium is facing upwards. So then we pin it and stretch it and visualize it with the microscope, this high spinning disc confocal microscopy microscope. Okay, so I hope that you can see. So this is how endothelial dependent hypopolarization looks with your naked eye. So for those of you who have never seen that, so here, that is actually the myoendothelial projection here. So you can actually visualize here the dense part. And notice when we introduce acetylcholine, notice what happened to the signals is that it went all crazy. It went like it gets faster. So there are more activation upon introduction of acetylcholine. Okay, so a certain terminologies that I would like to introduce. By the way, can you see the video clearly? I take that as yes. Okay. So there's a few terminologies that I would like to introduce is that we call IP3 calcium release as IP3 calcium pulsar. So because it pulses, so the kinetics of this, when they release the calcium, is actually very different from TRPV4. So TRPV4 we call sparklets because TRPV4 channel, you know, it is actually a cluster of 4 channels. So when it can be activated by 1, 2, 3, or 4, so it's like sparklets. It's like that. So that's where the terminology came from. So let's take a look at baseline. So what happened in both of this animal when there's no drug whatsoever in the system? So we can see here number of site of activation at baseline. It's the same. It's not significant. And then the number of site, the frequency is not significant as well. And the pulsar amplitude is actually our control. In order to determine that we are actually taking, you know, the action or the flashes, the signals, pardon me, the signals at an accurate point. So we have this as our positive control. Okay, so let's see what happened when we introduce ACH. As we, you know, the result or the signal is representative. You can see here. So every spike that you see right here is a representation of an open IP3 channel. Okay, here there's a very few spikes compared to the one in wild type. So this is the results from animal that cannot activate PKG via oxidant. To summarize that, you can see that baseline is both normal, and when you introduce, it doubles the site, the number of site whereby calcium is released is increased, and the frequency of the release of calcium is also increased, and our control is the same between the two, showing that we are recording the signals from the same from the same receptor. Okay, now we're going to take a look at TRPV4. The reason why we take a look at TRPV4 is just to validate the findings that we find in the previous functional experiment, the Zamiah graphic. So here you can see that even the duration, okay, sorry. So in order for me to see the TRPV4 calcium signal, so upon activation with ACH, because when you activate ACH, you activate both, isn't it? Pulsar and also, IP3 calcium release and also TRPV4 calcium influx. So in order for you to silent one, we incubate with CPA, and in order to release all the calcium within the ER, and then we minimally activate the TRPV4 channel by the activator. On top of that, we introduce ACH. So we can see here, there's a lot more calcium activity coming from the TRPV4 channels. Okay, and the duration is longer as well. Compared to here, it's long, but it's only 1 signal and 2 signals, and the rest is just silent. So the result is very representative of the thing, you know, the dilation that we see earlier on. And here, upon activation, you can see that the number of TRPV4 activity significantly increased. However, it is blunt, it is silent in the PKG animal, and the MEP sites and non-MEP sites, you can see there are so much more activity in the myoendothelial projection because TRPV4 is concentrated there. It is also it is present. We have detected it, and it is present in other places, not just in MEP, because TRPV4 channel is also sensitive to temperature. Okay. So let's just recap of all that information. So in health, this is the mechanism, a well-established mechanism that's been established. So we have ACH, GQ protein, and we will, in activation of all this, IP3 to the ER will cause a calcium release, and DAG will cause activation of TRPV4 clusters channel, and this would lead into a localized calcium increase in the myendothelial projection. So that would lead into activation of potassium channel, leading to hyperpolarization of the endothelial cells, and leading to the vasodilation of the artery. So what happened in the resistant mice, that we do see a significant decrease in calcium pulsars and then we can see a significant decrease as well in calcium release just now, and also no dilation to the direct activator. So that is why we don't see any dilation at all, or this explains why our by oxidant is hypertensive. So now we're trying to understand what we see in these animals. So in order to verify this in healthy arteries, what I'm going to do next is to introduce oxidant, and to see if it is actually, if it blunts this whole entire mechanism. And to our not so surprise, pardon me, I don't know what happened with it moved. Okay, let me walk you through this. So within the same animal, baseline, and then we have ACH. It increases the calcium pulsar. So this is the IP3 signals, yeah? It increases the calcium pulsar. So when I introduce PEG catalase, so PEG catalase is actually a permeable oxidant. So oxidant that actually can penetrate through cells. We can see a reduction. As soon as I put catalase on top of ACH, we can see a reduction here, not just in pulsar frequency, but also the number of sites whereby IP3 is reduced. So first we can see 40 number of sites of IP3, and then when we activate with ACH, we see 80 number of sites, it doubles. But when I introduce with oxidant, that can penetrate through the membrane into the cells, it reduces to 60. So this one is still quite preliminary. We are still trying to add on to the add on numbers to the experiment. So another way to look at it is to activate PLC in the oxidant resistant arteries. So if we know that there are something wrong here, so why don't, in the disease model, we activate phospholipase C and see if it works? So here is the baseline. The disease animal model is slightly higher pulsar frequency here, and then we have and then when we introduce ACH, for some reason it reduces. And let's take a look at this one. So, but when we introduce a PLC activator called M3M-FBS, it increases. It increases not just in the pulsar frequency, but we also see this increase in the number of sites of the pulsar frequency. Okay, so which means that the number of sites of IP3 receptor activation on the endothelial, on the ER is even more. Okay, so pharmacological inhibition of oxidant and direct activation of PLC, it shows us that oxidant activation of PKG is involved in the generation of BIP2. And this is down, so Since it is blunt, then it cannot generate PIP2, and that's why we're not seeing all this mechanism. So because now we know that there's something wrong here, but what is it then? There'll be, the next step is to look into immunology and a lot of molecular data, which with the interest of time, I would share, I would stop here, but we know that there is a biological, or no, it's not there is a biological, that oxidant activation of PKG acts as a biological anchor in order to activate PLC. Oops, sorry. Oops. So just a short conclusion that M3 induced endothelial vasodilation by acetylcholine is a highly conserved pathway, and PKG and oxidizable PKG have never thought to be involved. So this is a new mechanism that that we just unpuzzled. Then evidence shows that oxidant activation of PKG is critical in the function, in the dilatory function of endothelial in small resistant artery. So with all this data, like what's the rationale? Like why are we doing this? It's because, well, the generation of targeted oxidants to control calcium signaling in artery may represent a new therapeutic target to restore endothelial function. So, and as one of the key things that I would like to share with you today is that our collaborator in King's College London actually found the compound, which we call it G1 drug, with 93.7% accuracy, that this compound would bind to C42 region at the protein kinase G to form that disulfide bond to activate it. So there's, and I will be able to get it here and continue the research here. So, and why is this very important to look for another new mechanism? Is because there is an unmet need for a new hypertensive therapy, because this is from our national health survey. The updated ones is that only half of our population are aware that they are hypertensive, but that half, only 90% are on medication. But unfortunately, only 45% have their blood pressure controlled. So what happened to the remaining patients with refractory hypertension? And if it's uncontrolled, these people, our people are exposed to other comorbidities that would lead to even more complex clinical management in the future. Therefore, you can see that there's an unmet need for a new therapeutic approach, to understand, not just understand the underlying mechanism of hypertension, but also to find a new solution. Okay, and all of this could not have been possible for if I don't have this amazing team that working with me. So these are the teams of chemists that we're working with in King's College London. They have give us the minds and some of the expertise. This is our team here, vascular team in Manchester. In University Malaya. And here are the teams in Manchester that I've been spending 3 years with them, investigating small arteries. So, and before I end, I have another personal vendetta. I miss being in a very big, team and a very big group. So, my next research in I would love to look into CBD on TRPV4 channel. We have some preliminary evidence showing that cannabidiol, an extract from marijuana, shows a activity. It opens up TRPV4 channel as well. So we would like to dissect that mechanism and see if there's any potential for therapeutic use of cannabidiol in hypertension. So if any of you have interest in the therapeutic potential of CBD, or if you think that there is an application of endocannabinoid system in your field, please, email me, so that, we can always have a chat over coffee. And if you can email me by today, then I'll invite you to a talk on CBD tomorrow. That it's going to be an external talk. So I hope I'm not taking too much time. Thank you so much for listening. And before we end, I want you guys to take 3 take-home message. Okay, first of all, the take-home message is oxidant activation of PKG in the endothelium is vital, is critical for small artery vasodilation. So that is the the novel mechanism. So to answer the question, is it a friend or a foe? It's definitely a friend. And oxidant, a biologically produced oxidant, is definitely a friend. Okay. And also, the second take-home message is there's a new toy in the faculty, and if you think that pressure myography would help you in your research, if you want to try mounting, your patient's artery from any biopsy or any surgical procedure that you can obtain the artery, so come and hit me up and we can discuss about it. And also, if you would like to be a part of the CBD team, also email me. We can have a chat on that as well. So thank you so much, everyone, for listening, and I hope you gain a lot from my talk.
Okay. Thank you very much, Dr. Sharifah Zamiah. It's very interesting talk. Okay, you got like insight into our cardiovascular research, especially on the role of oxidants. Okay, so there are actually a lot of questions.
Oh, this is great. Okay.
Yeah, I got about 6 questions, but where couldn't find it? Wait,
I could not see the question as well.
Wait. Just now I got Q&A, but now the screen becomes small. Where is the Q&A? Hey, the organizer, where is the Q&A? Sorry.
Q&A button is at the bottom, on the bottom bar.
No, I couldn't. There's no, because just now I can see during the presentation, I can see the Q&A, but now there's no more. For
your Zoom,
chat,
Zoom application.
Where?
Okay.
Yeah, okay, fine. It suddenly becomes small. Okay, okay, I don't know if we can handle all the questions because of the time, but yeah, it's got around about 6 questions. I hope that you can answer it wisely so that we can at least to answer almost all the questions. So
Okay.
first question actually, what are the substances that cause oxidation of PKG? Do we know any currently?
Oh, the substance that causes oxidation of PKG?
Yeah.
So the substance is hydrogen peroxide. We do know. However, when we activate or when we incubate our cells with hydrogen peroxide, the formation of the disulfide bond it formed, but it is not as great as PEG catalase. So one of the things that a lot of people that works with oxidant do usually use is PEG catalase, because PEG catalase are able to permeate, able to penetrate through the membrane, so that it could activate PKG, because PKG is inside of the cells. So that there are 2, but however, physiologically, for healthy individuals like us, the oxidant actually is being detected from a mechanism sensor. So this is actually from high pressure, like interluminal pressure. So the higher your blood pressure, so the more oxidant that is being produced. So that data is it's already been published, and it's in the slides just now. Yeah.
Okay, thank you.
Okay.
I hope this is answered.
Thank you for the question. Good
So,
question, though. That's very important.
Yeah, really good. The second question is, we all know nitric oxide plays a huge role in the modulation of microvasculature as well. Okay, how does nitric oxide feature in your scheme of things? A parallel pathway, or has no interaction at all with this vasodilation pathway that you described?
So you're right. There are interaction of nitric oxide as well, but how am I supposed to? but it's been shown that the nitric oxide involvement in small arteries is not as great. It does not give a great dilation or as fast dilation compared to activating it via ACH. So that is one thing that we have seen in our lab. And one of the problem is because nitric oxide is such a sensitive, in order to activate that pathway is quite tricky. So there is a preliminary, I think it's already been published by Prof. Van Hout, showing that EDHF is so much more prominent in small arteries. But in our laboratory as well, we did the comparison. It's just that with nitric oxide, we're having a lot of problems. It oxidizes as well. It's not stable, and yeah. So and we see a faster dilation, a faster response with ACH.
Okay, thank you. So the next question, the link with nitric oxide and acetylcholine may be through nitric oxide synthase. So any comments on that?
Maybe the link with
nitric oxide.
There's a link. There is actually evidence of a link on that in large arteries, but in this research we did not even look it down to that angle. But you know what, that actually could be a really brilliant suggestion for our next experimental design to actually look into that.
Okay. The next question, what was used as an oxidant agent in the experiment?
What was used as an oxidant agent in the experiment is PEG catalase. So PEG catalase, the oxidant agent that's been used, are only used on the healthy animals, on healthy mice. That is just to verify that by introducing this oxidant, we don't see any activation or any signals any signals in the inositol triphosphate, the IP3, in any calcium release. So we tested that and we don't see any signals. So we tested that with PEG catalase.
Okay, good. The next question here, the autoimmune based antiphospholipase C is interesting. Okay, in the antiphospholipid syndrome, which causes thrombosis, could it be also due to vasoconstriction?
Oh, right. So Prof. Guna, such a great insight that this question. You know what? There are possibilities. Yeah, there are possibilities, but this is actually a bit, beyond my intellectual capability. I really can't, give you a confident answer at this point. But yes, to answer your question, yeah, there is possibility. But to explain on how that actually interacts, so you have to give me a bit more time. I'll probably have to email you back, Prof. Guna.
Okay.
Okay.
Another one, clinically, how could this intrinsic oxidant be activated?
Another brilliant question. So the only way it could be activated if you could prove that the G1 compound that we discovered, not that we discovered, my collaborator in London discovered, if we could prove that one, are not toxic and it works, then that could be the next potential antihypertensive medication. So yeah, but you know how clinical trial works. It probably it's probably going to take another 5 to 10 years. But yeah, but we are on it. We are definitely on it.
Okay, the last one, I think from this, hopefully the last one, in sepsis, okay, the first part of the cardiovascular system to be affected is the microvasculature, okay? So could it be due to a contribution of antioxidants in this arena?
Could it contribute to the contribution of antioxidants?
Yes. Yeah.
Sorry, just let me have a think.
So this is actually the contribution. Yeah.
Affected the, okay, so sepsis is the first part of the cardiovascular system to be affected. we introduce, yeah. When you say about antioxidants, there's a lot of antioxidant is actually very important for our system as well, and it has shown beneficial, properties on the vasculature. However, when we talk about oxidant, we're talking about oxidant induced due to a disease or a biologically induced oxidant. So to answer your question, to be honest, it could be due to, yeah. So I don't want to guess on it. But from my intellectual opinion, I think no, sorry, it could not be due to antioxidant in this area. No.
Yeah.
But that's actually something that we would take note on. Thank you so much though for the burning question. Anything else?
Yeah, there's I think it's gone.
Wow, it keeps on coming.
I cannot see. There's a lot of actually lots of
Really?
people interested with your.
These people
I
don't really
think
give me any rest.
For Zamri, there are some questions in the chat box.
Oh, okay.
Where's the
I
chat
think
box?
that's gone in Q&A. Any other question in chat box? Sorry. Okay, there's still got question here in chat box also. Sorry. Okay, would your findings be applicable to all parts of the body with small arteries, example the eyes? Okay, endothelial dysfunction is a major factor towards diabetic retinopathy.
Yes. Oh, brilliant question. You know what? Who asked that question? But that is a very brilliant question because ongoing at the moment, as we speak right now, that is being it's being designed. The experiment is ongoing. So it's not only that we're looking into mesenteric arteries, where we're looking into renal and also the eyes and other organs. But as for the interest of this research, I just present on mesenteric. But yes, to answer your question, yeah, we do look into other organs, and but we have not yet compared chondrite artery or tried to look into oxidant activation of PKG in chondrite arteries yet.
Wait, another one. Wait, it's moving so fast. Wait, is the PKG K channel pathway affected by oxidant concentration?
No, because I remember incubating 10 and 30, and both of them actually form a disulfide bond. So it's not concentrated. It's not concentration affected.
So this actually relates to the bigger questions, okay, of whether too much antioxidants can be detrimental and negate this physiological protective mechanism, right?
Yeah, that's right. And if you take a look at the clinical trials on antioxidant, I think there's a quite recent one that they run. I'm sorry, I can't recall what was the recent antioxidant, but there was one that they did with ascorbic acid, vitamin C at 1,500 milligrams, and the patients there's no improvement of blood pressure. But that is a clinical trial. People don't really try to delineate the mechanism, because before going to clinical trials, even though it is a known compound, it is always best to confirm the fundamental signs, to see and to witness with your eyes, and how does it really work before we push it to the clinical trials. So that's a very good insight on that.
Yeah, since we still have actually about 7 or 8 minutes to finish, so there is one more question, I think last from the participants. We could answer this. There are acute and chronic issues with respect to the vessels in the microvasculature. So your study seems to be associated more with the acute issues, right?
Yeah, you're right. Yeah, you're right, because we do see that the animal, where the PKG oxidation is silent, they develop hypertension at a very young age, and we start our experiment with quite a young mice, at week 16 to 20. So it is an acute representation of the disease. You're right.
Okay, great. So I think there's a one last question from me on the visualization of the calcium signals experiment. How can you tell if the signals are from the target desired receptor, the receptor that you desired? How can you actually tell if this actually from the signal from that target?
Okay, all right. That's a very good question, Rob. So the thing is, when we use the software, it is actually a customization. It's a customized software called Sparkend,
Okay, great.
whereby we set the signals. And before we set the signals, we did an electrophysiology experiment to identify, to actually, target the frequency and when the channel opens, and what is the frequency and also the kinetics of the channel. So that is prior to dysfunctional. We actually did a electrophysiology to identify this channel characteristic and the way of it opens and the sensitivity to the agonist that we use. So those experiments have been done prior to the functional experiment, just to confirm that we are looking at the same channel. They're not the same. We're looking at the correct channel. Yeah.
Okay, this is very interesting. Do you have that's the software here? Can continue to use the software?
Yeah. So the software is actually, so we have a teams of physicists, like people with physics to help us develop that software. I do have the software here, and if anyone would like to just take a look at how it works and how we detect. It's available. And the great thing is that it is automated, but even after automated detection, we still detect it with our bare eyes, because when the endothelium is actually split open, there's a lot of other factors, if the plane is moving, if the water is a lot, because train an experiment like that, you have to control everything to make sure that it is resemble of the physiological our body physiology. So that's why we double check our recordings automatically and also with our own bare eyes.
Okay, so we don't need to pay the license or whatever this for this software?
Well, I know. I don't think so. I mean, it's just
no need to pay any license.
But then again, I would probably have to ask the collaborators. You're right. Now that you mentioned it, but it is a customized software to our interest. Yeah.
Make sure we have a proper license or legal to use the software here.
It's the thing, because it is an in-house software, so we don't have any of that. They just keep it around.
Okay. One last question. Why did you choose mesenteric arteries in your experiments?
That's actually a brilliant question. I think mesenteric arteries is one of the best arteries to see the differentiation of conduit vessel, small artery, and resistance artery. Because if you take a look at the artery itself, you can tell that in the middle it's a conduit artery. The lumen is big, and then as it branches, that is the first order is slightly smaller, and it branches some more. And when you use third order resistant arteries from the mesenteric, you actually can see the capillaries kind of invaginate out from the vessels, from the resistant artery onto the lumen, onto the ileum. So it's a precise representation of resistant artery. So that's the first reason. And the second reason is basically so that we are able to compare our data, with other vascular researchers around the world. So that's why a lot of researchers prefer to use mesenteric arteries. Okay. So it's like a standardization.
Okay, great. Thank you very much. So I think we come up to the end of the session. So I would like to thank Dr. Sharifah Zamiah for very good presentation today, and all participants. Okay, there's a lot. I think we got around 60 over participants. So it's really good. Okay, we hope that you all really got very good, some new insights into the cardiovascular vascular research. And if you still have any other questions, probably you can contact Dr. Zamiah, or we call Mia in the department, okay, through email, whatever, or maybe we can have some like marijuana coffee with her.
You're gonna get me sick.
You mentioned the cannabidiol, so maybe you can have like marijuana coffee to have some more meaningful discussions, something like that. Okay.
That would be
So
great.
thank you very
so
much also
everyone.
for organizer for this opportunity for pharmacology department. And okay, thank you very much. Okay, bye.
Thank you, Prof, and thank you, everyone.